Next is the regulation of genes and their products, which is the Lac Operon. First, when we say gene regulation, it is the ability of a cell to control or regulate what proteins it makes from its DNA. When we say genomic DNA, it contains both structural genes, which encode products that serve as cellular structures or enzymes, and regulatory genes, which encode products that regulate gene expression. But before we start, let us tackle central dogma of gene expression as one important concept in this discussion. To keep it very simple, central dogma basically says that information is copied from DNA to RNA, which is known as transcription, and from RNA to proteins, which is known as translation. Moreover, we will use this concept when we discuss Lac Operon. Gene regulation of Lac Operon was the first genetic mechanism to be understood clearly. Now, if you look at the term itself, it is made up of two words, lac and operon. Lac meaning lactose, which is a disaccharide sugar present in a milk, and operon means a group. Basically, Lac Operon is a group of genes that regulates the metabolism of lactose. So this is the structure of Lac Operon. This is basically a small piece of bacterial DNA and consists of a few important genes that code for the enzymes that allows the bacteria to break down lactose. The Lac Operon can broadly divided into two important regions. First, the structural genes and the regulatory portion. The structural genes code for the enzymes, whereas the regulatory region of Lac Operon controls or regulates these structural genes. The regulatory region of Lac Operon allows the bacteria to essentially switch on or off the structural genes depending upon the lactose were available in or not. There are three important structural genes in Lac Operon known as LAC-Z, LAC-Y, and LAC-A. LAC stands for lactose and ZYA are arbitrary alphabets usually denotes the names of these genes. The region in blue is the regulatory portion of Lac Operon, and it is divided into many subregions. We have the LAC-I genes, which code for the protein repressor. We also have promoter and operator. We have our enzyme, RNA polymerase, that has to copy information of three structural genes of Lac Operon into mRNA. RNA polymerase always bind on the side located just behind the structural gene, and this is known as the promoter. In front of the promoter, we have the operator. You can remember that operator as a switch that can be turned on or off. When it is turned off, the RNA polymerase cannot move forward in the DNA. The genes can transcribe. When it is turned on, the RNA polymerase and the promoter can now move forward, and the genes LAC-Z, YA can transcribe into mRNA and then used to make protein. To use the lactose, the bacteria must express the Lac Operon genes which encode key enzymes for lactose uptake and metabolism. To be as efficient as possible, E. coli should express the Lac Operon only when two conditions are met. First, the Lac repressor acts as a lactose sensor. Second, catabolite activator protein CAP acts as a glucose sensor. Here is the illustration when lactose is absent. The Lac repressor binds tightly to the operator. It gets in RNA polymerase away, preventing transcription. With lactose, allolactose rearranged lactose binds to the Lac repressor and makes it let go of the operator. RNA polymerase can now transcribe the operon. CAP isn't always active. Instead, it's regulated by a small molecule called sialic AMP. CAMP is a hunger signal made by E. coli when glucose levels are low. CAMP binds to CAP, changing its shape and making it able to bind DNA and promote transcription. Without CAMP, CAP can't bind DNA and is inactive. This is catabolite repression. Low glucose. When glucose levels are low, CAMP is produced. The CAMP attaches to CAP, allowing it to bind to DNA. CAP helps RNA polymerase bind to the promoter, resulting in high levels of transcription. When the glucose is absent and lactose is present, the Lac repressor is released from the operator because the inducer, allolactose, is present. CAMP levels are high because glucose is absent, so CAP is active and bound to the DNA. CAP helps RNA polymerase bind to the promoter, permitting high levels of transcription. Here, no transcription of the Lac Operon occurs. That's because the Lac repressor remains bound to the operator and prevents transcription by RNA polymerase. Also, CAMP levels are low because glucose levels are high, so COP is inactive and cannot bind DNA. Furthermore, in this illustration, no transcription of the Lac operon occurs. COMP levels are high because glucose levels are low, so COP is active and will be bound to the DNA. However, the Lac repressor will always be bound to the operator, acting as a roadblock to RNA polymerase and preventing transcription. That's the end of my topic discussion for Lac operon.